Great. Well, thanks very much. Thanks for the invitation. Happy to present some of our results today. I've sort of structured my talk loosely around the R03 proposal that we got funded. And I'll say the title probably gives us a little too much credit. It's not really at all sort of a hypothesis-driven piece of work that landed us here. In fact, we kind of stumbled into the Dark Kinome. We were working with our colleague, Nathaniel Gray, at the Dana-Farber, working on sort of new chemoproteomic approaches to interrogate some of the covalent compounds that he was making in his lab. This is a snapshot of some of that work. Here we're taking his compound THC1, which is a CDK7 inhibitor. We're sort of elaborating it with an affinity handle to see if we could use this as what you might think of as a pharmacologic activity probe, and let the desthiobiotine tagged version compete with the native inhibitor and sort of get a very focused set of kinase targets or off targets using this approach. And the quantitative data are shown down here. We identify all these targets based exclusively on the cysteine containing peptide that the covalent inhibitor binds. You can see the acrylamide warhead here. And we throw all the other peptides away. So it's kind of a focused assay in this respect. And you see, worked very well. The controls here, CDK7, the positive or the primary target. Suspected off targets were CDK12 and 13, but this kinase kind of caught our attention, PKN3. We find this covalently modified on cysteine 840, and you see a nice dose-dependent competitive binding here. And we sort of knew we were in trouble. I asked Nathaniel, what about this kinase PKN3? And he said, I never heard of it. So we thought that sounded kind of interesting. You don't learn much about PKN3 from PubMed, but it has this, in Pharos, it's categorized as a T-bio, so not the darkest kinase, I guess, but still not a lot known about it. You learn a little bit more, or some other interesting data is present in the TCGA and other cancer genomics databases. You can see PKN3 is overexpressed in a range of solid tumors, in particular amplified in a decent subset of the more aggressive prostate tumors. And data from other groups published a while back seem to suggest that the kinase contributes to the metastatic potential in pancreatic cancer. So perhaps a reason to think about developing a selective inhibitor of this kinase. So at the time, pardon me, at the time, we had this molecule THC1, which of course bound PKN3 pretty potently, as I showed you, but this compound, because it is a very potent inhibitor of the cyclin-dependent kinases, it's not very good as a tool compound because it tends to be pretty toxic to a lot of cell lines. So we raided Nathaniel's freezer looking for other compounds in this family and came across this molecule JZ128. Had very little interest in the CDKs, but seemed to inhibit PKN3 fairly robustly and was well tolerated by cells. So in a whole series of biochemical experiments, just sort of a snapshot here, we were able to confirm that indeed it inhibited kinase activity. This is a covalent inhibitor, so we can disable the warhead and get a nice shift in inhibitory activity. And then biochemically, we were able to confirm that, in fact, this compound JZ128 did competitively inhibit PKN3 and really had no activity against the CDKs. Verified this with genetics in PKN knockout cells, in which we added back flag and HA tagged either wild type or cysteine to serine mutant kinase. And then this kinase is part of a small family, but it is the only one of the three that has this cysteine located proximal to the kinase domain. And so this compound JZ128 has no inhibitory activity against the other two family members. So overall, we were able to verify activity of this compound in vitro against PNK3. We're able to show that cysteine 840, which is how we identified this initially in our CITe-ld chemoproteomic experiment, was obligate for the pharmacologic activity of JZ128. We could see cellular engagement by JZ128, and as I say, we could confirm that it had no activity against the other PNK3 family members. So that was very encouraging. And this technique we developed, CITe-ld, as I mentioned, is a very focused assay, and this slide is just intended to sort of capture some of the value of having something that's a bit more focused. So here you see again this waterfall plot for JZ128. Now we're detecting 680 or so covalently modified cysteines, but the small subset here that show dose-dependent response, you see we capture both kinases and non-kinases as off targets here. And we compare this to the standard kinative assay. This turned out to be about a little under 300 kinases assayed here, and you see we capture everything that comes out of kinative except for this PIP4K site, which we later confirmed was due to the fact that this was in a peptide that's about 60 amino acids long or something. So we had to do a subdigest to capture this guy. But the point is, in contrast to these large cysteinome profiling experiments where the goal is to sort of assess, cysteines all across the proteome or capture all cysteines across the proteome and then figure out what's binding and what's not. When we compared our data from CITe-ld to these large compendia type studies here, what was quite interesting actually is the majority of sites that we found being competitively bind by these molecules, including this site on PNK3, were not even represented in some of these large compendia. So we thought it was a nice example of having these sort of tightly focused assays to characterize these compounds. So we ended up with a set of tools that, again, I have to say we sort of stumbled upon this kinase PNK3. And so we basically used our set of tools as kind of preliminary results to write up our small R03 grant. And these are some of the activities, experiments that we had proposed doing in the context of that grant. And one of the things is to go after substrates. And we heard Tudor, I think, mentioned yesterday some of these commentary type papers, that we're all sort of looking under the same street lamp, if you will. But one thing to keep in mind is that there's some collateral damage that results from all of these understudied kinases, which is outlined in this paper here from David James and Sean Humphrey, which is sort of this notion of a dark phosphoproteome as well. So they make the comment here that similar to the dark kinases, a huge proportion of phosphorylation sites are basically assigned to the top 20% of kinases. This is probably a little fungible because, of course, there's quite a bit of crosstalk and redundancy in some of these sites, but nonetheless, I think you see the point here. And so over the years, we've used quantitative phosphoproteomics in the lab with pretty good effect, I would say, to map in a very detailed way specific substrates. This is just one example of a pathway that we've been involved with over the years. This is understanding how dephosphorylation, in this case, of 53BP1 at a couple specific sites in the foci-forming region are required for the protein to shuttle into the nucleus and localize at specific sites of DNA damage. So we identified these two phosphorylation sites and mapped them to the activity of the phosphatase PP4. And then in subsequent work, we were able to show that this regulatory subunit of the phosphatase is phosphorylated near the C terminus. And we mapped that activity specifically to CDK5. So now, we're able to sort of map in great detail specific phosphorylation sites and substrate relationships in these pathways. And I would say we're reasonably effective at this. We've done this for a number of kinases now over the years, phosphatases as well. And more recently, we've actually sort of extended this to other target classes. So as a result of working with our colleague Sarah Burlich, who's a real thought leader in developing chemical probes to the deubiquitinating enzymes, we've been able to use some of these compounds now to start to identify specific DUBs that are responsible for deubiquitinating certain residues here, also in the foci-forming region. So we're sort of in a position now, as you heard mentioned from Sean Gomez, to sort of think about how, crosstalk amongst kinases and some of these other enzyme classes and how they regulate key functions in cells. None of these are understudied kinases, although I guess the doves are sort of understudied in their own right. This is just a snapshot of how we've tried to use then quantitative phosphoproteomics to start to map substrates of PNK3. And one of the nice advantages of working with the covalent probes is that you get chemical controls, so you can disable the warhead, and then you also have genetic controls. You can mutate the cysteine in this case to residues that will not be covalently modified. And now we have the advantage of having two different inhibitors, each of which has a fairly small constellation of targets, but they share only PNK3 as a common target. So this is really valuable for sort of knocking down the noise, if you will, in these large quantitative phosphoproteomic screens. So we do a large sort of TMT multiplex quantitative phosphoproteomic analysis here. We get something like 15,000 unique sites of phosphorylation, and the data are never black and white, so you have a lot of noise in data. And when you have more than one compound along with these controls, you can sort of map on each map the expectation of height of kinase and reduce. We were able to from about 15 sites to about 100, and further triage that and predict here, and we could get down to about 20 or so phosphorylation sites, some of which we've been biochemically validating. And then a shout out really quick to Allison Axton, because at a meeting she alerted us to the fact that they had identified this compound as another potent PNK3 inhibitor. And so we've been using that, setting up experiments to use this. And so we've been able to set these experiments up using a larger or new reagents that give you the ability to use a larger number of channels in these experiments. And I can't emphasize enough how important it is. Now we have 3 molecules which share one common target of PNK3. I think this is going to knock the noise down even further in these experiments. So really quite valuable. Even if these compounds, never see the light of day as a true inhibitor, as tool compounds, you can combine them in this way, and I think it's very, very powerful. So the other thing we were doing in the context of this grant was working with our colleagues in the Gray lab to optimize our compound JZ128. And of course, they kick these things out using this sort of modular mix and match strategy. They kick compounds out like it's a bodily functions. Now to start to carry, and we a little behind on this with the COVID shutdown back in the summer.
Jarrod, we need to wrap up.
Oh, sorry, the schedule said I was supposed to have 25 minutes. Is that right? Okay, that's all right. I'll wrap up. Sorry about that. Finish by saying the last thing we've got a small covalent fragment library. We're starting to use this to look for new chemical starting points for the Dark. Working with Gary Johnson to use his multiplex inhibitor beads as kind of an enrichment reagent so that we can incubate fragments in pooled format. And as I say, identify and validate some of these fragments as potential new starting points for the dark kinases. And I'll just end there and thank my colleagues, Nathaniel Gray, Sarah Burlich, and the others that are making these covalent probes that we've been able to characterize using some of these new chemoproteomic techniques. And Gary Johnson and others at UNC have been very helpful, and these are some really good collaborations that we're hoping to build. And this is the crew in the lab. Thanks for your attention. I'm sorry, I misunderstood the time.